A 71-year-old woman named Josephine came to me 20 months ago. She had been a secondary school biology teacher for 33 years. She understood cells. She understood DNA replication. She understood the difference between a controlled study and a testimonial. And she came to me not in panic and not in desperation, but with a folder of printed research papers and a question that she had been trying to get a satisfying answer to for the 6 months since her breast cancer diagnosis and her completion of the first phase of her oncological treatment. The question was specific. She had read about compounds in certain vegetables that had demonstrated anti-cancer activity in laboratory research. She wanted to know which of those compounds had the most credible evidence behind them. She wanted to know the mechanism, not the headline, the mechanism. She was a biology teacher. She knew that cells could be killed by many things that would also kill everything around them. She wanted to know whether the compounds she had been reading about were genuinely promising, or whether they were the nutritional equivalent of a compelling anecdote dressed in the language of science. She said, "I teach children to evaluate evidence. I want someone to evaluate this evidence with me, honestly." I sat with her for 45 minutes. I told her what the research actually said. What was laboratory data in cell lines, important but not the same as clinical evidence? What was epidemiological significant but confounded? What had moved into human trials and with what results? And which 4 vegetables had accumulated the most credible, multi-layered body of evidence for compounds with genuine anti-cancer biological activity that was relevant to a person eating real food in the real world? I'm going to tell you what I told her. I'm Dr. Kevin Adewale. I'm a cardiothoracic surgeon. I spent over 30 years operating on hearts, repairing arteries, and watching the relationship between what patients eat and what their cells do over decades. What I'm about to share with you, most doctors won't bring up at your annual checkup, but after 60, it matters more than you think. Today, I am going to walk you through the 4 vegetables with the most credible scientific evidence for compounds that support the body's cancer defense mechanisms. What those compounds are, what they actually do inside the cell, what the research shows and where its limitations are, and how to eat these foods in the way that maximizes their biological activity. Stay with me, because the 3rd vegetable on this list has a specific preparation requirement that determines whether the active compound is present at all. And most people eating it are destroying that compound before it ever has a chance to work. Before I take you through the 4 vegetables, I need to be precise about something that most health content on this topic fails to be precise about. Because imprecision in this area is not just intellectually frustrating, it is potentially dangerous. It can lead people to make decisions about cancer prevention or cancer management based on a misunderstanding of what the science actually shows. And I will not do that to you. There is a meaningful difference between 3 categories of evidence. First, laboratory evidence. Studies showing that a compound kills cancer cells in a petri dish or slows tumor growth in a mouse model. This evidence is real and it is important. It is the necessary starting point for understanding biological mechanisms. But a compound that kills cancer cells in a petri dish may be ineffective, unstable, or toxic at the concentrations required to produce the same effect in a living human body. The gap between laboratory activity and clinical benefit is where most promising compounds fail. Second, epidemiological evidence. Population studies showing that people who eat more of a certain food have lower rates of certain cancers. This evidence is also real and important. But population studies cannot establish causation. People who eat more broccoli also tend to exercise more, smoke less, drink less, and make other dietary choices that independently reduce cancer risk. The broccoli may be the active ingredient, or it may be the marker of a pattern of behavior that is doing the protective work. Third, clinical trial evidence-controlled studies in humans showing that a specific dietary compound reduces cancer incidence, slows progression, or improves outcomes. This is the highest standard of evidence, and it is the category in which the evidence for dietary vegetables is most limited, not because the compounds do not work, but because the clinical trial infrastructure for studying dietary interventions is fundamentally different from and less well-resourced than the infrastructure for studying pharmaceutical compounds. I'm going to tell you what the evidence at each of those 3 levels shows for each of the 4 vegetables. I am going to tell you where the evidence is strong, where it is promising but preliminary, and where it should be interpreted with caution. And I'm going to be honest with you about the difference between those categories because Josephine deserved that honesty, and you do too. Think of it this way. The body has multiple layers of cancer defense. DNA repair mechanisms that correct errors during cell replication. Tumor suppressor proteins that monitor cells for abnormal growth and initiate programmed cell death. Apoptosis when they detect it. Immune surveillance systems that identify and eliminate cells displaying abnormal surface proteins. Antioxidant systems that neutralize the reactive oxygen species that damage DNA and initiate the mutations that can lead to cancer. And detoxification enzymes in the liver and in other tissues that process and eliminate carcinogenic compounds before they can interact with DNA. The compounds in the vegetables I'm about to describe work primarily by supporting, enhancing, or activating one or more of those existing defense layers. They are not chemotherapy. They do not kill cancer cells the way a cytotoxic drug kills cancer cells, aggressively, indiscriminately, with deliberate toxicity. They work by making the body's own defense systems more effective at doing what those systems were already designed to do. That distinction is important because it reframes the expectation from a miracle cure to a biological support system. And the biological support system is genuinely worth understanding. The first vegetable is broccoli, and the specific compound is sulforaphane, a molecule produced in broccoli when the vegetable is chewed, chopped, or otherwise physically disrupted. I want to describe the mechanism in some detail because sulforaphane's mechanism of action is one of the most well-characterized of any dietary compound with anti-cancer properties. And understanding it changes how you think about what the vegetable is doing inside your body. Here is what is actually happening inside the cell. Broccoli contains a precursor compound called glucoraphanin and an enzyme called myrosinase. They are stored in separate cellular compartments. When the broccoli cell is damaged by chewing, by chopping, by blending, the compartments are disrupted and glucoraphanin and myrosinase come into contact. The enzymatic reaction converts glucoraphanin to sulforaphane. Sulforaphane enters the cell and does something specific and remarkable. It activates a protein called NRF2, nuclear factor erythroid 2 related factor 2, which is a master regulator of the body's antioxidant and detoxification response. When NRF2 is activated by sulforaphane, it migrates into the nucleus of the cell and switches on hundreds of genes involved in antioxidant enzyme production, detoxification enzyme production, and anti-inflammatory signaling. The cell becomes more resistant to oxidative DNA damage. Its detoxification capacity for carcinogenic compounds is enhanced. Its inflammatory signaling, which, when chronically elevated, creates an environment in which cancer cells can initiate and proliferate, is reduced. Additionally, sulforaphane has been shown in laboratory studies to inhibit histone deacetylase enzymes, HDACs, that are involved in silencing tumor suppressor genes. In cancer cells, HDAC activity is frequently elevated, suppressing the genes that would otherwise initiate apoptosis in the abnormal cell. Sulforaphane's HDAC inhibition partially reverses that silencing, allowing tumor suppressor genes to be expressed again. The epidemiological evidence is substantial. A meta-analysis published in the European Journal of Nutrition found that higher cruciferous vegetable consumption was associated with significantly lower risk of several cancers including colorectal, lung, and breast cancer, with the protective association being most consistent across studies for cruciferous vegetables specifically, rather than total vegetable intake. The cruciferous vegetable family, which includes broccoli, Brussels sprouts, cauliflower, kale, cabbage, and rocket shares the glucoraphanin-sulforaphane pathway. Broccoli has among the highest glucoraphanin content of the family. The clinical trial evidence is smaller but genuinely promising. A study from Johns Hopkins University, where the original research on sulforaphane and NRF2 was conducted, found that sulforaphane-rich broccoli sprout extract reduced the levels of aflatoxin DNA adducts in people in a high-risk area for liver cancer, aflatoxin being a potent liver carcinogen, by approximately 50%. That is a direct measure of carcinogen DNA interaction. The sulforaphane was intercepting the carcinogen before it could damage DNA. The specific preparation point I mentioned at the beginning applies here. Broccoli must be chopped or chewed to initiate the myrosinase reaction that converts glucoraphanin to sulforaphane. But here is the preparation detail most people miss. High heat destroys myrosinase. Boiling broccoli for more than 4 to 5 minutes inactivates the enzyme. If the enzyme is inactivated before the glucoraphanin has been converted, sulforaphane is not produced. The precursor compound is present, but the conversion does not happen. The solution is either to eat broccoli raw, to steam it briefly, no more than 3 to 4 minutes, or to chop it and allow it to rest for 40 minutes before cooking, giving the myrosinase time to complete the conversion before the heat deactivates it. Alternatively, adding a small amount of raw broccoli, raw mustard seeds, or raw radish, all of which contain active myrosinase to cooked broccoli, rescues the sulforaphane production because the external myrosinase completes the conversion that the heat destroyed in the broccoli itself. That specific detail, understood at the cellular level, changes how you prepare the vegetable. And that change determines whether the compound responsible for the biological activity is present in the food you eat. The second vegetable is garlic, and the compound allicin follows the same precursor enzyme disruption pattern as sulforaphane. Allicin is produced when allicin, stored in garlic cells, is converted by the enzyme alliinase upon cell disruption. Crushing or chopping garlic initiates the conversion. Heat inactivates alliinase. Exactly as heat inactivates myrosinase in broccoli, the same preparation principle applies. Crush the garlic, wait 10 minutes, then add it to cooking. The 10-minute window allows allicin production to complete before heat deactivates the enzyme. Here's what is actually happening inside the body when allicin and its metabolic derivatives, diallyl sulfide, diallyl disulfide, adenine, reach the cell. These organosulfur compounds have demonstrated multiple anti-cancer mechanisms. In laboratory research, they inhibit the formation of nitrosamines, carcinogenic compounds formed in the stomach from dietary nitrites, by scavenging the nitrogen intermediates before they can react. They enhance the activity of detoxification enzymes in the liver, increasing the rate at which carcinogenic compounds are metabolized and eliminated. They have demonstrated the ability to induce apoptosis in cancer cell lines through multiple pathways, including caspase activation and cell cycle arrest at the G2M phase. And they inhibit angiogenesis, the formation of new blood vessels that tumors require to sustain their growth through inhibition of VEGF signaling. The epidemiological evidence is consistent and geographically replicated. The Iowa Women's Health Study found that higher garlic consumption was associated with a 50% lower risk of colon cancer. Research from the Linxin region of China, an area with high rates of esophageal and gastric cancer, documented significantly lower cancer rates in population subgroups with higher garlic consumption. A meta-analysis published in the American Journal of Clinical Nutrition found that higher allium vegetable consumption, garlic, onion, leeks, shallots, was consistently associated with reduced risk of gastric and colorectal cancer across multiple population studies. The clinical evidence includes a controlled trial published in the Journal of the National Cancer Institute that found garlic supplementation reduced the number and size of colorectal adenomatous polyps, the precancerous lesions that precede colorectal cancer, compared to placebo over 12 months, that is a step closer to clinical relevance than most dietary compounds achieve in controlled trials. 1 to 2 fresh cloves daily, crushed, rested for 10 minutes. Added to food last rather than first in the cooking process to minimize heat exposure. That is the preparation that preserves the biological activity. That is the difference between eating garlic and eating garlic with its active compounds intact. Tell me in the comments, have you been cooking garlic directly in the pan without resting it first? Because that single change, 10 minutes between crushing and cooking, is the preparation detail that determines whether the allicin is present or absent in the food you put on the plate. The third vegetable is the one whose preparation requirement I told you would surprise you. And it is the one most people are preparing in a way that eliminates the active compound entirely. Cooked tomatoes, not raw tomatoes. Cooked, this is the reversal of the broccoli and garlic principle, and it is where people get dangerously confused about what processing does to food's nutritional activity. The compound in tomatoes with the most credible anti-cancer evidence is lycopene, a carotenoid pigment responsible for tomatoes' red color, unlike sulforaphane and allicin, which are produced by enzymatic reactions that heat destroys. Lycopene is made more bioavailable by cooking. The cell walls of raw tomatoes trap lycopene within a fibrous matrix that the digestive system cannot fully break down. When tomatoes are heated, particularly when heated with a fat, such as olive oil, the cell walls are disrupted, the lycopene is released from the matrix, and its absorption from the gut into the bloodstream is significantly enhanced. Cooked tomatoes deliver 2 to 8 times more bioavailable lycopene than the equivalent quantity of raw tomatoes. Here is what lycopene does inside the body at the cellular level. Lycopene is one of the most potent antioxidants in the carotenoid family. It quenches singlet oxygen, one of the most reactive and DNA-damaging forms of reactive oxygen species, more efficiently than beta-carotene or alpha-tocopherol. In cell biology, singlet oxygen-mediated oxidative damage to DNA is one of the initiating events in the DNA mutation sequence that leads to carcinogenesis. By reducing the concentration of singlet oxygen in the cellular environment, lycopene reduces the probability of oxidative DNA damage. Beyond antioxidant activity, lycopene has demonstrated specific effects on intercellular communication through gap junctions, the channels that allow adjacent cells to exchange regulatory signals. Cancer cells frequently lose gap junction function, which is part of how they escape the growth regulatory signals from surrounding normal tissue. Lycopene has been shown in laboratory studies to restore gap junction communication, making cancer cells more responsive to the growth inhibitory signals of their neighbors. The epidemiological evidence for lycopene is strongest for prostate cancer. The Health Professionals Follow-Up Study, one of the largest and most methodologically rigorous dietary cohort studies, found that higher tomato product consumption was associated with significantly reduced risk of prostate cancer, with the association being strongest for aggressive and advanced prostate cancer rather than indolent forms. A review published in Cancer Epidemiology, Biomarkers and Prevention confirmed the association across multiple cohort studies. The clinical trial evidence is smaller. A pilot trial published in the Journal of the National Cancer Institute found that lycopene supplementation in men with prostate cancer awaiting prostatectomy produced measurable reductions in PSA levels and reduced the incidence of surgical margin involvement compared to placebo. 2 to 3 servings of cooked tomato products weekly, tomato sauce, tomato soup, roasted tomatoes prepared with extra virgin olive oil to enhance lycopene absorption. That is the preparation that delivers the bioavailable lycopene the research is measuring, not raw tomatoes on a salad, though those provide fiber and vitamins of their own. Cooked tomatoes with olive oil for the lycopene that is the subject of the cancer prevention evidence. The fourth vegetable is spinach, and the broader category of dark leafy greens, specifically for 2 separate and mechanistically distinct anti-cancer compounds, folate and chlorophyll. I am addressing them together because they are most concentrated together in dark green leafy vegetables. And because their mechanisms are complementary rather than redundant, here is what is actually happening inside the body with folate. Folate is essential for DNA synthesis and repair. Every time a cell divides, it must replicate its entire DNA, approximately 3 billion base pairs, with minimal error. Folate is a cofactor in the biochemical pathway that produces the building blocks of DNA, specifically the thymidine and purine nucleotides that constitute the genetic code. When folate is deficient, DNA synthesis becomes error-prone. Uracil is misincorporated into DNA in place of thymine. The DNA repair machinery detects and attempts to correct these errors, but the correction process itself can produce strand breaks and chromosomal damage. Folate deficiency therefore increases the rate of DNA damage and the probability of mutations that initiate carcinogenesis. Research published in the Journal of the National Cancer Institute found that lower dietary folate intake was independently associated with increased risk of colorectal adenomas, the precancerous polyps that precede most colorectal cancers, and that the association was particularly strong in people who consumed more than one alcoholic drink per day because alcohol directly impairs folate metabolism and absorption. The folate-alcohol interaction in colorectal cancer risk is one of the most well-characterized gene-nutrient lifestyle interactions in cancer epidemiology. Chlorophyll, the compound that gives dark green vegetables their color, has a separate and more recently characterized anti-cancer mechanism. Chlorophyll and its derivative chlorophyllin form tight molecular complexes with polycyclic aromatic hydrocarbons and aflatoxin, two of the most potent dietary carcinogens, in the gut, reducing their absorption and their delivery to colonic mucosal cells and to the liver. A controlled trial published in PNAS, the Proceedings of the National Academy of Sciences, conducted in Qudong, China, where aflatoxin exposure from contaminated grain is a significant liver cancer risk factor, found that chlorophyllin supplementation reduced urinary aflatoxin DNA adducts by 55% over 3 months. The chlorophyll was intercepting the carcinogen in the gut before it could reach and damage the DNA of target cells. Dark leafy greens, spinach, kale, Swiss chard, rocket, watercress are the primary dietary sources of both folate and chlorophyll. The preparation principle here is moderate cooking, or raw consumption. Brief steaming or sautéing retains most of the folate content while reducing the oxalate content that can be problematic in large raw quantities, particularly relevant for adults with any kidney function concerns. As I discussed in the video on kidney disease and diet, a large handful of dark leafy greens, daily raw in salads, lightly steamed as a side dish, incorporated into soups and stews, added at the end of cooking to preserve folate content. That is the practical target, not a supplement. The whole food, with its full matrix of fiber, micronutrients, and phytochemicals, produces interactions between its components that isolated folate supplementation does not replicate. Let me bring all 4 together, because what makes these 4 vegetables specifically worth discussing is not that any single one of them is a cancer cure. None of them is. Cancer is a complex, heterogeneous group of diseases driven by genetic, environmental, and biological factors that no dietary intervention alone can prevent or treat. That is not a caveat. That is the truth. And the truth is what Josephine needed from me and what you deserve from this video. And what makes these 4 vegetables worth discussing is the quality and the mechanistic specificity of the evidence behind them. Broccoli and the Cruciferous family sulforaphane, activating NRF2, enhancing cellular detoxification, inhibiting HDAC enzymes that silence tumor suppressor genes. Garlic allicin and organosulfur compounds, inhibiting carcinogen formation, enhancing liver detoxification, inducing apoptosis in cancer cell lines, inhibiting angiogenesis. Cooked tomatoes, bioavailable lycopene-quenching singlet oxygen, restoring gap junction communication associated with reduced risk of aggressive prostate cancer in human cohort studies. Dark leafy greens, folate supporting accurate DNA replication and repair, chlorophyll intercepting dietary carcinogens in the gut before they reach target tissue. 4 compounds, 4 mechanisms, 4 vegetables with preparation methods that determine whether those compounds are biologically active in the food on your plate. Knowledge without action is just worry. I'm not here to give you a list. I am here to give you a mechanism and a plan. I call this the Anti-Cancer Dietary Support Protocol. 5 practical steps beginning this week. Step 1. Add 1 cruciferous vegetable to your diet daily. Chopped and rested for 40 minutes before cooking, or lightly steamed for no more than 4 minutes, or eaten raw. Broccoli, Brussels sprouts, cabbage, kale, cauliflower. Rocket. Rotate through the family across the week. The glucoraphanin content varies across the family, and variety provides the broadest sulforaphane exposure. Step 2. Crush 1 to 2 cloves of garlic daily. Rest for 10 minutes. Add to cooking at the end rather than the beginning. Make that 10-minute rest a structural part of meal preparation. The biological activity of the garlic depends on that window. Set a timer if necessary. The 10 minutes is not optional. Step 3. Cook tomatoes with extra virgin olive oil at least twice a week. Tomato sauce. Roasted tomatoes. Tomato-based soups. The combination of heat and fat is the preparation that delivers bioavailable lycopene. Replace raw tomato garnishes in at least 2 meals per week with a cooked tomato preparation using olive oil. Step 4. Eat a large handful of dark leafy greens daily. Spinach, kale, rocketea, watercress, Swiss chard, raw in salads, briefly wilted in warm dishes, added to soups at the end of cooking. The folate and chlorophyll content make this the simplest and most consistent anti-cancer dietary habit available from a single food group. Step 5. Bring these dietary additions to your next oncology, GP, or specialist appointment. If you have a cancer diagnosis or a family history of cancer, ask your oncologist or GP specifically whether any of these dietary additions interact with your current treatment or medication. Cruciferous vegetables can affect the metabolism of certain chemotherapy agents through cytochrome P450 interactions. That is a conversation to have before making significant dietary additions to an all-active treatment plan. Not instead of making the additions, but in coordination with the clinical team managing your care. Dietary intervention and medical treatment work best as partners, not as alternatives. I want to come back to Josephine. She implemented all 4 changes within the first week of our conversation. She became, by her own description, meticulous about broccoli preparation. She rested her crushed garlic for exactly 10 minutes. She had a kitchen timer she used for nothing else. She made a tomato sauce with olive oil twice a week that she described as the best unintentional recipe she had ever developed. And she ate rocket and watercress every day with a lemon and olive oil dressing. At her 6-month oncology review, her oncologist noted that her inflammatory markers, CRP and interleukin-6, were significantly lower than they had been at her post-treatment baseline. Her oncologist did not attribute this solely to diet. Multiple factors contribute to inflammatory marker reduction. But the oncologist did note that Josephine's overall metabolic health had improved measurably and that the dietary changes she described were consistent with the anti-inflammatory trajectory of her blood panel. She came back to see me 7 months after our initial conversation. She brought 2 things: her blood panel and a research paper she had printed and annotated with the precision of a 33-year biology. The teacher who had read it carefully and wanted to discuss its methodology, she placed both on the desk. She looked at the blood panel first. Then she looked at me. And then she said the thing that has stayed with me ever since. She said, "I'm a biology teacher. I taught children for 33 years that mechanism is everything. Understanding why something works is the only reason you ever truly believe it will." She paused. Then she said, "You gave me the mechanism, and the mechanism gave me the conviction to do it consistently." That is the difference between a health instruction and an education. Instructions are followed until they become inconvenient. Mechanisms are followed until they become unnecessary. Because once you understand what sulforaphane does to NRF2, you do not need willpower to rest your chopped broccoli. You just understand what happens if you do not. Which of these 4 vegetables are you already eating, and which preparation change will you make this week? Tell me in the comments. I read every single one, and those answers help me understand what the conversation about diet and cancer prevention is still failing to communicate in the places where most people receive their health information. If this video gave you something that changes how you prepare food this week, share it. Not for me. For the person in your life with a cancer history, or a family history, or a clinical risk factor who has been told to eat more vegetables without being told which ones, in which form, prepared in which way, for which specific reason. They may not know that the 10 minutes between crushing the garlic and adding it to the pan is the difference between active allicin and a flavoring, but you do now, and that is exactly why you are here. And if you have not subscribed yet, please do. Every week I bring you honest, evidence-based information that I wish every one of my patients had access to long before they needed surgery. Take care of your heart. It has been working for you every single minute of every single day without asking for anything in return. Give it what it needs. It is a fair trade. One last thing, and this is critical. Everything I have shared today is general educational information based on published research and my clinical experience as a cardiac surgeon. It is not a substitute for your personal medical care. It is not cancer treatment. It is not cancer prevention in any guaranteed sense. If you have a cancer diagnosis, please do not make dietary changes without discussing them with your oncologist first, particularly if you are receiving active treatment. Some dietary compounds interact with chemotherapy agents, and that interaction must be assessed by the clinician managing your treatment. The vegetables I have described support the body's natural defense mechanisms. They do not replace medical treatment for established cancer. They know your diagnosis. They know your treatment plan. I do not. This is education. Use it as the beginning of that conversation